So in this video, we're going to explore our first type of secondary structure, the beta sheets. So let's begin this by drawing out an amino acid that's part of a peptide. So we start with the N-terminus, we go through our alpha carbon carbonyl, peptide bond to another nitrogen, alpha carbon carbonyl, peptide bond nitrogen. We now go through alpha carbon, another carbonyl, and we keep this up. Okay, so let's go ahead and stop here and point out that this side of the peptide is the N-terminus and the other side is the C-terminus. We know that because within a single amino acid we have the nitrogen written first and the carbonyl written last. I'm going to go ahead and now draw a second peptide that's oriented just above this one, and this time I'm drawing from the N-terminus to the C-terminus. But this time, the N-terminus is written on the right, and the C-terminus is written on the left. So what you'll notice here is some very obvious places for hydrogen bonding between our two backbones. So the carbonyl oxygen hydrogen bonds with the hydrogen on this nitrogen. And we end up with these very strong hydrogen bonds between the oxygen and the nitrogen. This orientation of two peptides with opposite polarities is known as anti-parallel beta sheets. So we have one peptide going from the N-terminus to the C-terminus, and the one that's hydrogen bonding with goes from C-terminus to N-terminus. And when these line up in this anti-parallel fashion, we have these very strong hydrogen bonds between them, making this a very stable beta sheet. The other orientation for a beta sheet is known as parallel, and that's when the two peptide chains, which I'm drawing out here, are oriented in the same direction. So this first one I have drawn from N-terminus to C-terminus, and the second one I'm drawing here, you'll note, is drawn in the same direction from the N-terminus to the C-terminus. When we draw out the polypeptide chains in this way, we still have hydrogen bonding, but now as you can see, the hydrogen bonds are not as lined up as well. And when they're not lined up as well, they're not as strong. So this orientation of the beta sheet we call parallel, because they're oriented in the same direction, both of them from N-terminus to C-terminus. And when this happens, we get weaker hydrogen bonds because they're not lined up perfectly. So just to review here, we compare the parallel orientation to the anti-parallel orientation of our beta sheets. And remember, with anti-parallel, the polarity of our two peptide chains are opposite. So one goes from the N-terminus to C-terminus, and the other one from left to right is C-terminus to N-terminus. When we have this orientation, we have much stronger hydrogen bonds because they line up better. So let's go ahead and think about one more aspect of beta sheets. Remember when we were talking about the properties of peptides, we noted that sequential amino acids have side chains that alter from up to down. Well, the same thing goes when those peptides are part of beta sheets. So the first side chain comes out towards you and the second one goes behind the plane of the computer screen here. So because of this, we can very much predict the properties of beta sheets just like we can predict the properties of individual peptides. And the key to this really is remembering that we have alternating side chains. So one goes out, the other one comes back towards us. The next one is out, the next one back towards us. So if we put this into the context of a sheet, what I'm drawing here is this square that's meant to represent the two-dimensional properties of a beta sheet. This one is made up of two different peptide chains that are hydrogen bonded together to make an anti-parallel beta sheet. We think about the side chains. The first one goes above the plane, the next one below, the third one above, and the fourth one below. The same is true for the other chain. The first one above, the second one below, the third above, the fourth below. So we can use the arrangement of individual amino acids to predict the properties of this polypeptide chain.